So let's move on to the second in the series, The Role of Immunological Variation by Jeremiah Faith. Welcome.
Oh, Jesus.
You can do live commentary. You can just give us talk from the seats. Yeah.
Okay. Is that good? It could be interesting if you kept it. It'd be like a live color commentary like you get in the football games. So thanks, everyone, for coming. Is Vince Young here? Okay. So if you don't like the idea of putting human microbes into mice, you're going to be sick to your stomach. So, I like what you do. Okay, well, let's see. Okay. Any more than I know what I do, right? So it could be interesting. Hopefully we can have a bit of a discussion. So I think in terms of immunology, to me, the biggest publication to date is still even of 2009 from Cell. This is from Dan Lippman's group, where they showed that Jackson mice lack SFB and they have low Th17 cells. And then they showed pretty definitively that if you transfer an SFB microbe that's commonly in taconic mice, that you get this increase in Th17 cells, which is shown as you move towards the right and you add SFB. You can see the number of black dots in that little box increases, which shows that Th17 cells are going up. And I think this is important for two reasons. One, it showed that a specific microbe can change the immune system on top of a complex background. But also it's important for some topics that Andrew McPherson mentioned earlier, which is that it's very distributable. So all of us can buy taconic mice versus Jackson mice, and we can do this type of experiment. And it's made it the most common comparison, I think, in the immunology community. So just to give one example, there's many different disease models now where people have tried Jackson versus taconic, and they almost always find some type of difference. This is from 2015, where they did anti-PD-L1 treatment on these mice. But just look at the blue line versus the red line. The blue is a skin melanoma. And you can see that the melanoma in blue from taconic mice grows faster than in red from the Jackson mice. Again, two different microbiotas. The differences between these two microbiotas are quite drastic, but just shows that you get two different growths. And the sort of ability for us all to pay $20 or whatever it is to get these two different microbiotas in has made it that these are the two most common microbiotas. But then the question is, and this is something that we talked about a lot at dinner last night, what is a Jackson microbiota? What is a taconic microbiota? And hopefully, I think Craig might talk a bit more about this later. But I'm going to take a brief aside into some technology development. We like to build toys in addition to studying the microbiome. One thing that we're really keen on doing is switching the field to absolute terms. If you look at the microbiome now, most of the papers look at relative abundance, which is on the left here. These are our standard rectangles of different color that are higher or bigger rectangles means more absolute abundance, or relative abundance rather. But if you look at the top here, this is a fake example where we're using an antibiotic to deplete some microbes. So we're removing the yellow ones and the brown ones, and if you look in relative terms, it looks like a bloom of green bacteria. But in absolute terms, you can see that we're just removing the yellow ones and the brown ones, right? So the way you look at the data can change your thoughts on it. And the reason I'm explaining that is because, so this is being done by actually Eduardo Contajoca, MSTP student in our group. So the reason I'm showing this is because the way that we start with this is just by buying labs from Jackson, buying mice from Jackson Labs, which it's a pretty standard thing to do. And we just gave them vancomycin. The idea here was to deplete the microbiota to look at the absolute microbiome. And so that's done at time point 0. At time point 0, we give them oral vancomycin in their drinking water. And you can see towards the end, by day 7 or 13, you get this expansion of the firmicutes, which are shown in yellow. So if we then plot this in absolute terms, we're just going to plot the load of microbes in the intestine. What you can see is when you can't see the numbers so well, but at time point 0 is right when, or actually day 1 is when the microbiota collapses. This on the y-axis is how much microbes we have, and once we add the vancomycin, the microbiota kind of disappears. And so in absolute terms, if we scale it, it doesn't look like a bloom of firmicutes. It looks like the decimation of the community, right? So a pretty different way of looking at it. So we sort of started to use vancomycin as our standard antibiotic because it's quite good at depleting the microbiota. So if we wanted to check some effect on the immune system, we wanted to remove microbes, we would use vancomycin. But one day we bought some mice from Jackson Labs. Again, the same, put in the order, C57 black 6, they should be genetically mostly identical. And then this time the biomass did not go down. So we gave them vancomycin, but this time the biomass stayed more or less the same. And you can see now on the absolute microbiome plot on the right that there is a bloom. There's a bloom of Akkermansia, which has been mentioned multiple times. So to sort of sum it up, two mice from Jackson Labs, which has become this standard. We're going to compare Jackson versus Taconic. But depending on the day you buy the mice, the room you get them from, they're not the same microbiota, right? The only real quantifiable difference between these that we have right now is SFBs in Taconic, and it's not in Jackson Labs. But all the other parts can be so different that if you give antibiotics to two different batches, you'll get different results in the absolute microbiome. So this brings me to the concerns for animal welfare. And I think Andrew mentioned a bunch of this more eloquently than I can, but I just do it again. So the current standard is this idea of Jackson versus Taconic, or different animal facilities. But they're not standardized or defined. And the thing that worries me is that, if people came to work in our group and then they leave and they can't repeat what they did without bringing the animal facility with them, then I think it's almost not scientific. It creates, I think the idea of doing this heterozygote breeding is good because at least within a paper you can know that what they did was rigorous. But the best scientists come from things like E. coli, from inbreeding mice, from C. elegans, where we can all share the tools and we can hack on them together. And of course we need to go outside, like the idea of looking at rotten apples in C. elegans or whatever it was yesterday. Provides you a way to look at diversity, but also allows you to come back to a clean system. So because of that, we do quite a bit of gnotobiotics. And so this is the gnotobiotic facility at WashU. I should take a picture of ours, but it's in Manhattan. It's more crowded. And it's spread apart in a bunch of different rooms. But more or less, we have the same type of, I think, but just vertically distributed. But the old model of gnotobiotics was that you would have one isolator and one microbiota. And it becomes extremely difficult to do this if you want to screen many different microbial communities. So back when I was a postdoc, we started doing little hacks to see how well we could get one microbiota into one cage. And like almost everything when I was a postdoc, this was actually done a bit with Federico, where we like to just go mess around with things. And we found out that if we dealt with the cage very carefully, we could maintain germ-free mice for a little bit, about 2 weeks, like Balfour mentioned. But if we colonized them with the microbiota, we could maintain more or less the same community. And if we knew what we were putting in there, we could use next-generation sequencing to track and see if it was still in there. So when you go from one microbiota to one cage, the scalability goes way up. And I think since that time, there's also been these sort of advances in isopositive caging systems that you can now buy devices to help you do this. But I think those are quite good, and they're better than what we have here, a lot more costly. But the most important factor is not so much the cage as the person that's doing it. The way we train animal facilities now is to keep bad things out, whereas this is really trying to keep whatever's in there inside. So I think it takes a different level of SOP. So a lot of what we do now is sort of screening microbiotas with this main idea, like what's the difference between your microbiota and my microbiota, and how does this influence physiology? And by doing it with the same microbiotas, it means that if we have a student that goes and builds their own lab, we just have to hand them a tube of the microbes, and they can start from where they left off, not start from scratch. So Graham Britton's a postdoc in the lab doing quite a bit of this work, often in collaboration with Eduardo. So one simple experiment you can do that I think is potentially useful therapeutically is to look at fecal transplants. So as you probably know, fecal transplants are getting popular in the context of C. difficile. And this is a pretty standard plot which people show. This is PC1 is just the measure of sort of the difference between microbiotas. Here's the donor microbiota. Here's the recipient, which is far away from the donor, which just means that its microbiota is very different. And then when you give them the transplant, it comes down and looks more like a healthy person, so it clusters with the donor. So by doing this type of experiment in human, you essentially can do it one time with no replicates. The nice thing about gnotobiotics is you could then take that human microbiota, put it into several donor mice, put the recipient into several recipient mice, and then transplant the donor onto the recipient. So you can have multiple copies of this, and you can try perturbations to the fecal transplant to try to understand how it works. And you can see that, at least in this very extreme example, they look quite similar. Another thing that we worry about is the sort of manufacturability. So I've seen plenty of interesting microbiotas from stool that had a cool phenotype. And the ones that have the most interesting phenotype, the stool gets less and less and less because people use it so much. So one thing that we wanted to be able to do is to manufacture microbes. So we have a large anaerobic koi chamber in the lab. Inside of it, we have a robot. And then we can use this to manufacture the same communities. So we take a person's fecal microbiota, we use some mass spectrometry to dereplicate it, and we store them in 96-well plates. And we store these in such a way on-site and off-site that hopefully we have more or less an infinite supply. We can always make more of it if we want to. So it takes about a week to make one of these from a person. So we have in the freezer probably 40 or 50 different people where we've isolated as many microbes as we can from the microbiota. So one example of doing this in sort of this idea of standardization, we started trying to use the T-cell transfer model. And so although I'm giving a talk about immunology, I don't know a heck of a lot of immunology at this point, but I'm better than if I gave the talk 2 years ago. So when I first started, I didn't know any immunology, so I just went around and asked all the immunologists, what mouse model should I use for colitis? And everybody was suggesting this CD45RB high. This is the T-cell transfer mouse. They said, but if you want to do it, if you want to have colitis, you need to do it in this building over here, because if you do it in this other building, it doesn't have very severe disease. This is another thing that came up at dinner last night, that people get different phenotypes and it can change over time. So to sort of get around that, we want to know, how do the microbes in different people induce colitis in this model, more or less severe? But we needed a standard. So one thing that we've been doing is Graham manufactured one microbiota from one person. Each of these lines is just a different batch. Either we regrew it and made another cocktail of it, but this is all culturable, all repeatable. So this is the course of probably at this point, about 18 months of work. So every time he has a batch of mice, he's going to try to see how different human microbes manipulate this mouse model. He throws in a few mice with this particular donor's microbiota so that he can have a standard amount. So here on the x-axis is time, and over time they start to drop weight as they get sick. And this also correlates with histological damage and inflammation. And by having this standard in each batch, we can know, if we get a really strong deviation of this microbiota from the standard line here, we know that perhaps something went wrong with this batch. So every batch of animals, we stick it in this donor, but then we're also screening many different donors. So here, each line is a different human microbiota, either cultured or uncultured. And each line corresponds to about 6 animals. So it's the average of a series of animals. And what you can see is, based off the person's microbiota we put inside there, we can get either they grow perfectly fine or they have colitis and they start to lose weight, right? And the thing that you can do here with humans that you can't do with mice is that you can't go around and ask a mouse if they have IBD. I guess you can look at some inflammatory markers, but it's hard to screen many mice microbiotas because it's hard to know where to grab them from. What's the true mouse microbiota? But at least with humans, we can sample a number of them. And so it allows us to do things like split them up by colonizing with IBD microbiotas versus no IBD. So in red are the individuals with inflammatory bowel disease whose microbes we transfer over, and blue are the individuals without inflammatory bowel disease whose microbes we transfer over. And what you can see is that there's some healthy individuals that their microbes are able to lead to more severe disease. But overall, there's a strong separation between microbes transferred from people with inflammatory bowel disease versus those without. So that's the context of disease, but I think we always have this feel. I think the most frequent question at microbiomes talks is what's normal? So rather than having normal, we're just trying to look for multiple healthy individuals and see the distribution in immune profiles. So maybe none of us are normal. We're all sort of on this spectrum, just like we are genetically. So these are just different human microbiotas. These are the amount of FOXP3 positive, CD4 positive T cells, so Tregs. And you can see some people's microbes are able to induce quite a bit, some induce less. And there's always this distribution when we try multiple people's microbiotas. This is the same thing with Th17 cells, some very high inducers, some low inducers. So one thing that we're interested in doing is having consortium that are like our standards. And then once we find these nice distributions and differences between them, start to go in and find what are the specific microbes inside there that matter for the phenotypic effects. So the first time we did this was back when I was a postdoc. This was in collaboration with Philip Ahern, who's a postdoc in Jeffrey Gordon's lab. And we put 5 different people's microbiotas and mice, and here's germ-free. And you can see all of them induce regulatory T cells, shown here on the y-axis. So every person's microbiota was increasing T cells, which was actually shown by McPherson earlier today as well. And so we said, let's try to find out which microbes are doing this. And so we just take the library of microbes from someone, and we start to break it apart using ideas similar to what Jeffrey Gordon showed yesterday, trying to figure out which of these specific microbes are the one that increase regulatory T cells. So when we were doing this early on, we got a little bit worried that it seemed like many microbes were able to do this, and this seemed a bit different from what people would have guessed. But one important result that came out along the same time was that this paper that was mentioned earlier from Andrew McPherson's group, where they had put altered Shadler flora in the mice. And as you can see, and as he showed earlier, each of these is a different strain of mice. And when you go from the top, you go from germ-free to colonized. And going from top to bottom doesn't matter which mouse, it always increases regulatory T cells. So this paper was important because rather than looking at conventionalization of a complex community, it showed that only 8 microbes that were very well defined were capable of boosting regulatory T cells. And it showed us that it might not be such a big complex community that can boost regulatory T cells. So with that in mind, we started to screen for microbes that could do this. And we were able to identify several different microbes, about 7, that by themselves, monocolonized, could increase regulatory T cells relative to germ-free animals. So sort of continuing this on forwards, we started to move now in trying to identify other microbes that can modulate the immune system. One of these is Th17 cells. So this is an individual with Crohn's disease whose microbe, when you put them into unchallenged mice, increase Th17 cells compared to a healthy control. And if we compare our distribution of all the people that we've screened so far, you can see this individual lies on the extreme spectrum. And this gives us some type of feel for how we're going to go in and try to find this microbe, because we know whatever microbe this is somewhat rare in the population, so we can design our search strategy around that. And so to do that, Graham Britton took it was a 16-member community, and he started to do orthogonal groups. So he did 8 different groups shown by each column here, where each group has a different subset of the microbes. And in only 16 different combinations, he was able to identify the microbe that was doing this. So here's the p-value associated with each microbe and its ability to increase Th17 cells. You can see here there's a single organism, Enterobacteriaceae isolate 1, that's able to boost Th17 cells. And so each dot here is the communities where this microbe is present, and the minus sign means that the microbe was absent from the community. And then the nice thing about defined communities is you can start to really dissect this out. So he can take the entire community, just remove that one microbe, or he can take the entire community and remove a similar strain, but not identical strain. Or excuse me, similar family, I guess, but different strain. And both of those do what you would expect. And at the same time, we can create sort of very small defined communities of microbes from outside of this community. So this is a 7-member model community that you could buy from ATCC, similar to the idea that Andrew was talking about earlier of standardizing a community. Unfortunately, we haven't standardized with each other. Otherwise, this result would be more interesting. But nonetheless, we can take this 7-member community, colonize on top our microbe, and increase Th17 cells. Okay, so in terms of challenges for the future, I think most of this was addressed this morning. We didn't come up with a definitive solution, but I think my feelings are very similar, that SPF is designed to keep bacteria out of a community, to keep bad things out. But really, if we want to control the microbiota, we have to know what's in there. So I would promote the idea of developing standard operating protocols that would allow you to keep microbes in for a certain number of generations, similar to what Balfour was talking about earlier. How long can we keep a defined microbiota in a mouse if we have people that know how to properly do that? And the goal then would be that we'd have a certain number of microbiotas that have different properties that you could order from any vendor or from some sort of place that would be pre-colonizing your mouse, and you'd have it on F2 or whatever generation you wanted to. Again, logistically, this is complicated, but I think more important than anything, you would need a change in your SOPs and your training of the animal staff to be able to handle this. And then finally, I would like the idea of effector strains that you would put on top of it. You'd have defined microbiotas, and then you'd have these effector strains that can push different immune properties around. And if we all had the same baseline communities, we would know how good each effector strain is at invading a certain number of communities. So if Skip finds a new virus and we want to understand how well that virus can manipulate some immune population, it'd be nice to know how robust it is across the standard defined communities. So we need to build systems to do this, suppliers, vendors, and it needs to be something that we can manufacture and culture efficiently. This is my group. As I mentioned, quite a bit of this work was done by Graham Britton and Eduardo. The Treg work was done as a member of Jeffrey Gordon's lab with Philip Ahern. Got many microbiotas from many amazing physicians, and thank you for your time.